Hi, this is Dr. Fry, and this is my second lecture on the genetic problems. Um, so, last time we did complete dominance. That is when the dominant allele is completely dominant over the recessive allele, and you have two phenotypes. Codominance is where you they're codominant, and so you will have this is basically when you have patchy. An incomplete dominance is when it is not completely dominant over the small w, and so you will get pink here. So this is more of a blending, and this is more patchy. This time we're going to do multiple alleles, sex-linked, and pedigrees. And I'll show you one example of multiple alleles, and that would be human skin color, right? So there's three genes here that are working, A, B, and C, and there's probably more than that. But you see, the more dominant alleles you have, the darker your skin is, and the more recessive of the alleles have, the lighter your skin is. So all of these sort of work together so this one is heterozygous for all three genes, and that one's right in the middle. So that's an example. Another example is Labrador retrievers, and this is also an example of epistasis, which I'll show you in a minute. But black is dominant over brown, right? So if you're looking at the first one, he only has to have one big B to be black. The second allele could be a big B or a little b, it doesn't matter. And this doesn't really matter for him to be black. He does have to have one big E. And it doesn't matter what that second allele is. This one has to have recessive. So chocolate is recessive to black. This actually shouldn't be here. And he does have to have one big E. So the only way you get a yellow lab is if this gene here has two recessive alleles, and that over trumps this one right here. And that's what epistasis is this, when this is homozygous recessive, it doesn't matter what these alleles are, the coat color is going to be yellow. Okay. So this is what they did. So I'll just show you this. So this is a dihybrid cross, and they cross two heterozygous parents, and these are black, right? So these are both black labs because they have a big B, right? And they have a big E, and they're heterozygous. And I will go ahead and show you how they got those genotypes, these genotypes here, right? So this is a parent, this is diploid. It has two copies of each for each gene, or two alleles for each gene. Here they only have one allele for each gene. So I'll show you how they did that. So basically the parent is a big B, little b. Oops, I'm sorry. And erase that. And big E, little e. And so the alleles that can give here, he can give a big B or little b, and they can give a big E or a little e. And now you're going to do all those combinations. So this is big E, that's right there. And this one is going to be big E, little e, which is right there. And then you're going to have little b, big E, right there. And then you're going to have little b, little e, which is right there. And so you're going to get, whenever you have a heterozygous parent and you have you're looking at two traits, you're always going to have four different types of gametes. And since the mom and the dad are the same, this is the dad, and the mom is going to have the exact same alleles. And so we're going to talk about what the phenotypes are. And so for black, you're going to have big B, big B. You can have big B, little b. And you can have big B, big B. You can have so you can basically have 4 different genotypes to get a black, but it has to have one big B and it has to have one big E. And it really doesn't matter what the other ones are. So that should match up with those 9. You should have 9 of these, right? You should have 9 black dogs. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9 black dogs. And then for chocolate, you have to have a little b, but then you can, and you have to have at least one big E. Those are the 2 for the yellow, for the chocolate lab. And then for the yellow, it really doesn't matter what the first 2 are. So you can have a big B. It has to be little e. Not big B, little b. And that would be the genotypes for the yellow offspring. So, and I think that is right. So yeah, so that's they have all 3 of them there, and you can look at them or you can figure them out for yourself. Right, but that's the answer to that one. Okay, so on the cross picture, what must be the genotypes of 2 parents for the outcome to produce at least one yellow Labrador puppy, but no chocolates? And again, these are the genotypes that have to be. So you don't want any chocolates. So you could actually so in order for them all to be black, the parents are black, right? So they have to have at least one. They probably have 2 big B's because you don't want any chocolates, and chocolates are little B's. And the parent and you want yellow, right? But you also want black. All right. So you could make these. So you could do something like that. So this would produce no chocolate. So these are all going to be black because you don't have any little B's. And you can't have 2, you have to have at least one big E for to have a black lab as well. So I think this is the answer here. So this is the answer here. You could probably come up with another version of that. Okay. So 2 black labs have a litter of puppies. Out of 16, 8 were black, 3 were brown, and 5 were yellow. The sire's genotype was heterozygous for the B locus and heterozygous for the E locus. The dams genotype was found to be heterozygous for both traits as well. Construct a dihybrid Punnett square to show the possible genotypes. And again, I will show you how I got those alleles. And this is where a lot of people actually mess up. So both parents are heterozygous for both traits. So you're going to have a big B, little b, big E, little e. And so this parent can give a big B or a little b, and a big E or a little e. And now you're going to do those combinations. So this is big B, big E. And this is big E, little b, big E, and little b, little e. So I actually can't put them up there now. So I'm going to put them up here. So maybe it'll let me do it. This is going to be big E. And I didn't put the other one on there. Okay. All right, so let me go back to this. So now I'm going to go to here. This is going to be big E, big. And both of these parents are the same, so they should be the same. Right, and then you're going to do these crosses here. So I have to do them in here with this, I guess. All right. So that's going to be big B, big B. Big B, little b, big E, little e. This is going to be big B, little b. Okay. So what ones must be black then is, I'm going to do, I'm going to get rid of this. But we have the crosses now. So the ones that will be black, I'm going to do this gray. So these will be black. So it has to have a big B and a big E to be black. So this one's going to be black. This one's going to be black. It has to have a big B and a big E. All right, so I think that's all of them. So I should have 1, 2, 3, 4, 5, 6, 7, 8, 9. So I should have nine black pups here. And then brown, you have to have 2 little B's and a big E. 2 little B's and a big E. So I'll make this one brown. Oh, I didn't want to do that. So 2 little B's and a big E, that would be this one right here. Two little B's and a big E would also be this one right here. And it would be also this one right here. All right, so you should have 3 brown dogs. And then the yellows, it doesn't matter what the B's are, but they're going to be little E's, right? And here you are going to have 4 yellow labs because these all have little E's. So that's that problem there. All right, so these are multiple alleles, and this is blood type. So you can have blood type AB, or 0. And these are the phenotypes here, right? But you can have 6 different genotypes. So for type A, you can have AA or A0. Type B, you can have BB or B0. Type AB is just type AB. And type 0 is 00. And if you look at this up here, a lot of times they will put them with I's, but generally we drop the I's and just use so A and B are codominant, but I is actually little I, which is 0, is actually recessive. Okay. Okay, so we'll do a couple problems here. A man who has a type B gene, so he gave you the genotype. So the genotype here is going to be B, and is married to a woman with type O. So type O, she has to be a 00 or an OO. What blood type will their children have? So this is going to be a okay. So okay. So this is going to be a B0, B0, Okay, here we are. All right. So this one is going to be a this one's going to be a B0, a B0, and a B0. All right. So that means that, so what blood type will their children have? Their children will have a B0. They don't really have any choice. So, okay, we'll go to the next one. A man with blood type AB. So he has blood type AB. So I'll put the man on top, A, B. Is married to a woman with type O, so she's going to have 0, 0. And they have 2 natural children and 1 adopted child. The children's blood types are A, B, and 0. Which child was adopted? So you'll have to do this cross. So they can have a child with A, and they can have a child with B, but they're not going to have a child. They can't have a child with 0 because 0 would be, the genotype would be 0, 0. So the child that was adopted would be 0 because they can't have a 0, 0 here. Okay. So let me. Okay. So a type 0 has to have the genotype 0, 0, and they could not have a 0, 0 child. Okay, so I just showed you one example of that epistasis, and that was the idea that this yellow lab, so if this is double recessive, it sort of overtakes whatever allele is here at the B gene. And that's what this is also showing you here in pigmentation of mice. So the wild type is agouti, so that's AA. So this is this tan color here. And the recessive color, it would be little a, that is black. But then when gene C is recessive homozygous, that's CC, it negates the A gene, and the mouse is albino. So all of these should have homozygous for the little c. And that's exactly what we saw with the yellow lab. So these would be the yellow labs, basically. And these would be the brown labs. And then the rest of them would have been, the black labs. So, so epistasis means sort of standing upon, like, so it takes it over, right? And they are generally homozygous recessive, those traits, when they do that. Okay, so the next thing we're going to do is sex-linked. And basically, they're considered autosomal if they're carried on the first 23 pairs. So the first 23 pair are these. And the 23rd pair really determines your sex. And this is an XY, and you know that's a Y because it's not as big as the X and it's missing some. So a female would be XX. She would have 2 chromosomes that look like that. And an XY, the Y is missing part of the X, is basically what it's doing. So the first 22 chromosomes are autosomal, and the last one is sex-linked. And this is what we're talking about, is this right here. This is when we talk about sex-linked, we're talking about this pair of chromosomes. And so eye color in Drosophila, which are fruit flies. So wild type is red. So here's wild type. And recessive is white. And this is white here, but there's all kinds of blending here. And the males are homozygous because they only have one X. So this trait is carried on the X chromosome, not the Y. So it only has one X chromosome, so it's only going to have one allele. Okay, so we're going to do the following cross. And you have to keep track of the X's and the Y's here. So I guess I'm going to actually have to draw this, I think. All right, so I'll do the first one here. So this is the female. So she has 2 X's. And he is going to have an X and a Y. But the traits are carried on the X. So she's going to be heterozygous. And he only has 1 X, so that's going to carry that way. So then you're going to do that cross. This is going to be a female. Oh, I would just draw these. Just draw them, I guess. So this is going to be X, big R, X, big R. This is going to be X, big R, X, little r. This is going to be X, big R, Y, X, little r, and then Y. Okay, and then the genotypes. So R is dominant. So the genotypes you're going to have I did that. All right, so the genotypes you're going to have so you're going to have a an X. I'm just going to have to put the R's big. I guess I'll draw it. So you're going to have, I'll make it smaller. X, big R, and X, big R. And that's going to be a red-eyed female. So that's going to be a red female. Oh, gosh. That's going to be a red female. And then this one is going to be also a red female. This one is going to be a red male. And this is going to be a white-eyed male because that's a small r. So you're actually going to have all 4 of those. All right. So that's an example of sex linked. You have to keep track of the X and Y's on the sex linked. The last thing I'm going to do is pedigrees. So pedigrees are used to analyze the inheritance of particular traits throughout a family. Pedigrees show the presence or absence of a trait as it relates to the relationship among parents. So generally, this is what they look like. The females are round, the males are square, and generally this is the one with an affected trait. This is if they died. This is twins. This is adopted. Anyways. And then this is the first line. This is the second generation, things like that. So this is showing you this, right? So this is a female and this is a male, and these are married, and they had 3 children, and they had a male, they had 2 males, and they had a female. And then this female married this guy, and they actually had 4 children, and 2 of those are female and 2 of those were male. And this is if it has the trait, right? So and if it's recessive, you can have carriers, and carriers is basically when they're heterozygous. So that shows you that part. And probably only thing I'm going to ask you is to sort of pick out what kind of disease it is. So it can be autosomal, and again, autosomal is those first 23 chromosomes. It's on those first 23 chromosomes, which was here, right? So these first 22 pair of chromosomes are autosomal, and the last one is sex-linked. And that's what this, or X-linked or sex-linked, it's the same thing. Okay, and this is the pedigree of Queen Victoria. And so this is what this looks like, and this is hemophilia. And so this is a normal male, a carrier female, which means she is heterozygous. So only the females are going to be heterozygous because the males only carry one. This is sex-linked. And so this is a normal male, and this is a male with hemophilia. And you'll notice only males actually get this, right? So they're all squares that have hemophilia, and that's because it's sex-linked, and they only need one allele. They only need that one allele from their from the mother to get it. So to be a female to get it, you have to have two recessive alleles. But since the males they only have one X, so they only get one allele. So this is actually a sex-linked trait, and it's sex-linked and it's recessive. I'll get more to that down here. Okay, so there's 4 types. There's dominant and recessive, and then there's autosomal and sex-linked. So autosomal is if they're not on the sex chromosomes, which I just showed you. That's the first 22 pair of chromosomes, and sex-linked is on the X chromosome, and that's on the 23rd pair. Okay, that's on that 23rd pair that I showed you. And there it is again, right? So these are all autosomal, and this is the sex link. So this is an XX, that's a female. This is an XY, that is a male. So I'm just going to show you these, and I think all you have to do is sort of pick them out on the sheet. So autosomal means it's not on the sex chromosomes. It refers to those situations in which a single copy of an allele is sufficient to cause expression of a trait. So a big B. So it needs a capital letter. And so every person should have at least one affected. Every affected person should have at least one affected parent. It doesn't skip generations. So this is affected, right? So you can see there's black at every generation. So it does not skip a generation. Males and females should be equally often affected. An affected person has at least a 50% chance of transmitting the dominant allele to the offspring. So here is a affected parent, and that's 50% of his offspring. Here's an affected parent. It affected almost 50% of the offspring. Unaffected parents can have unaffected children because if they're heterozygous, they can have unaffected children. So you'll sort of look for these things when you look at these pedigrees. Examples of autosomal dominant would be progeria, in which the person ages very rapidly. Huntington's disease, in which the central nervous system starts to break down at the age of 30. So those are all examples of autosomal dominant diseases. The next one is autosomal recessive. And so this is where you have to have 2 recessive alleles to have that trait. And this is where you're going to have carriers. So, right. So here they made them a different color, but a lot of times they'll just have them like half black and half white. So that show that they're heterozygous. So we'll go over this one. So, an affected parent may not have affected person may not have affected parents. So right here, this affected child, these 2 affected children do not have affected parents, but both of those parents were carriers. So the disease can skip generations. So it's actually only in this one disease here, but it could actually show up in the next generation if this one would also marry a carrier. It affects both sexes equal, and it can compare to skip generations. Two affected parents will have affected children 100% of the time. So we don't have an example of that here because these are carriers, they're not affected. Autosomal recessive genes is albinism. So it's a genetic condition with the loss of pigment or hair. Tay-Sachs is the genetic disorder, which is a buildup of fatty deposits in the brain, eventually proving to be fatal. Sickle cell anemia and cystic fibrosis. So those are all autosomal recessive traits. The next one is sex-linked or X-linked. So this is recessive. So this refers to those situations where a recessive allele on the X chromosome can lead to a trait, condition, or disorder. So here the female will need two recessive alleles to carry the trait, but the male only needs one recessive allele because he only has one X. And that's why males are more affected than females. So disease tends to transfer from mother to son because she's going to give the only X for the son, and father the daughter, because if it's a daughter, he can actually give that X chromosome. And homozygous recessive females can arise only from matings from which the father is affected and the mother is affected or a carrier. And so here you also, since it's recessive, you're going to have carriers. So these are carriers, these are heterozygous. And you'll notice here that you are having, that you have two sons that are affected. So again, this is going to affect males are more affected than females. So the females a lot of times will be carriers and they can have affected sons, but the sons generally are more affected because they only have one X and therefore they only need one. If that allele is recessive, that gets expressed. Okay, so, some examples would be hemophilia, which is the inability of the blood to clot properly, muscular dystrophy, and color blindness. So color blindness generally is found more in men than women because it is X-linked. And this is X-linked dominant. So this refers to a situation where a single dominant allele on the X chromosome can lead to a trait or condition. So you don't have carriers when it's dominant. If you have a capital letter, if you have a dominant allele, it gets expressed. And here, twice as many females are affected as males, because again, you only need that one allele. Usually half the children of affected female will be affected regardless of sex. All the daughters of an affected male will be affected, but none of the sons, because the son won't get that allele. So, so here the male is affected, and he has actually affected all of his daughters. So, and you can see more females are actually affected than males. So here you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 females affected, and 1, 2, 3, 4 males affected. So that's showing you that one there. So vitamin D rickets disease is an example of that. And this is Y-linked. So the trait is carried on the Y chromosome and it is transmitted from father to son only. So if it carries on the Y, the females are not affected. So the father will affect all of his sons. So he gave that son this gave the Y to this son, and he gave the Y to this son. This dad gave the Y to this son, and he gave the Y to these sons. Right? So this dad was unaffected, so he didn't give it to his sons. So, and examples of this would be hairy eels, retinitis, and this. That's that. So I think, okay. Okay, so I'll skip that. So I will give you one more example, and this is really the Blue people of Kentucky. Um, so they're called the Blue Fugates. So this is a family that lived in the hills of Kentucky, of eastern Kentucky, and they're notable for having been carriers of a genetic trait that led to the disease, this right here, which gives sufferers a blue tinged skin. And so you can see here that the dad is blue, and they have 4 blue kids, right? But the mom is normal. This kid's normal. This kid's normal, and this kid's normal. So, the idea is what? So she has to be a carrier, right? So she has to be a carrier this is a recessive trait. It's autosomal recessive. So she has to be a carrier in order for these kids to get it. And it is autosomal because males and females get it. So this is a female. I'm not sure what this one is, but here's a female that doesn't have it. They have dress them in dresses, so it's hard to tell. Yeah, this is a female. This is a male. This is a male. This is a male. But one female has it, so you think it would be autosomal. It could be. So, all right. So that's it. So I think I went over all this, and hopefully that was helpful.